Everybody, and welcome to our departmental grand rounds all around San Diego this morning. Faculty and trainees are waking up and saying, is it Monday or is it Tuesday? And is it grand rounds today? But I'm sure they'll be wandering in with a confused look on their face. I'm just here for the one purpose of introducing Amanda Barkley Levinson, a postdoc in Abe Palmer's group, to do the speaker introductions.
Great. Good morning. Thank you for all being here. Today it's my pleasure to introduce our speaker, Dr. Yasmin Hurd. Dr. Hurd is professor of psychiatry, neuroscience, and pharmacological sciences, as well as the Ward Coleman Chair of Translational Neuroscience at the Icahn School of Medicine in New York. Her translational research program examines the neurobiology of drug abuse and related psychiatric disorders. She leads a multidisciplinary team of investigators where they use molecular biology, behavioral neuroscience, genetics, and neuroimaging techniques to study the human brain as well as translational animal models. She combines this preclinical research with clinical laboratory investigations that evaluate the therapeutic potential of novel medications in the treatment of substance use disorders. Dr. Hurd has been extremely productive in her career. She's published over 160 articles and reviews. She publishes consistently in high-impact journals in her field, and her work has also been covered in the popular press. She has a long track record of NIH-funded research, and she currently has 3 R01 awards from National Institute on Drug Abuse. In addition to this research, she also has served on numerous professional society committees, NIH study sections, and editorial boards. And she's received many awards and honors to many to go into right now. And then on top of all this, she also serves as the director of the Addiction Institute within the Mount Sinai Behavioral Health System, which covers one of the largest addiction populations in the US and provides clinical care supported by science-based medicine and advanced state-of-the-art research. Today, she'll be presenting some of her work in her talk entitled Molecular Insights of the Human Heroin Brain Provides a Path to Novel Treatments. We're very glad to have her here, so please join me in welcoming Dr. Hurd.
Thanks, Amanda. Thanks very much. I have to say, when I saw the schedule, like 8 o'clock, each day from opioid use disorder or overdose. And, it depends on which region where they may have naloxone available to try and save people, so the numbers will fluctuate in terms of the number of people dying. But it basically, it's a huge crisis. And we know that many of the people who have now gone into heroin, they started off abusing opioid pain prescription medications, where we still have too many prescriptions being, too many of these pain prescriptions being written for the population of the US, with over 200 million of these. So, I was put this number up, and everybody, it's shocking in part still that drug overdoses, like over 70,000 people died. It was 2017. The same thing was for 2018. And it peaks all of these deaths related to the peak of the HIV deaths, car accidents, even gun violence in the US, which is really a horrific number that we've gotten there. And what's more is shocking for me still is just by the numbers that when you calculate over, for example, the past, I've done this number at that time of over the past 17 years of how many people died of opioid overdose. nearly 700,000 people, and it exceeds the number that died in conflicts and wars in the US from 1941. That's, I mean, again, people don't get the impact, even though they see it in the news um, all the time. But for every person that dies, so all of those overdose numbers are horrific, but there are actually many more people who are surviving with this disorder that need treatment. And in fact, um, opioid overdose or opioid abuse or opioid use disorder, they cost more, 3 times more than other medical disorders to treat. So they'd estimate like over 400, about $500 billion dollars. So at Mount Sinai, this has been a big thing for us because we have over about 6,500 people who are in our opioid use treatment programs, and over 10,000 people with chronic pain disorders that we're trying to also provide treatment for and decrease their addiction vulnerability. So as most of you know, most of the medications that are used for treating opioid use disorders are opioids themselves, such as methadone, buprenorphine. And the question for us is definitely on a scientific level, it's been, you know, can we start coming up with alternative treatments? And I'm going to go back to say the reason that for the alternative treatments is that one of the things the opioid epidemic really emphasized is that the medications that are currently used to treat opioid use disorders, they have such governmental regulations and clinical burden. So I said to you, we have over 6,000 people that we treat with in our with methadone for the most part, buprenorphine in a smaller percentage. But you have to watch 6,000 patients. And so, very long time ago, definitely a masochist, decided to try to see if we can do molecular studies in the postmortem human brain, where I was like beaten up for trying that. But it's really helped a lot because it's helped to provide us some insights into what is the molecular profile in people who have been abusing opioids for a while. So this is looking at gene expression in the nucleus accumbens of people who died of heroin overdose. And you don't need to be a great computational biologist to see that the gene expression pattern in control subjects differ from those in heroin abusers, even though we do pay our computational people a lot to do this. So that led us to a lot of down a path that I didn't expect, because when I started studying addiction, dopamine was, you know, one of the main transmitters that with addiction, it's dopamine. But when we started looking at the heroin abusers and opioid system, obviously, one of the things that was really clear was that most of the gene dysregulation, you could say, was really related to two major systems. One was the glutamatergic system, and particularly relation to synaptic plasticity. And a number of animal models in addiction obviously had emphasized glutamatergic disturbances in addiction. But another one that was clear was epigenetics. And for those of you, you know, I'm not going to, sometimes we talk about the genetics of addiction. You know, we're talking about complex disorders, and genetics definitely plays a role into vulnerability, hands down. But a major aspect of addiction, as many complex disorders, are the environmental impact on the DNA and so-called epigenetics, in a sense of studying the mechanisms of gene regulation that doesn't really account for changes in the DNA itself or the DNA sequence, but in terms of often the structure, whether or not a gene is turned to regulate how a gene is turned on and off. And there are many different types of epigenetic mechanisms. There are, excuse me, I'm still fighting. Well, the flu won, I lost, but I'm still fighting. Still, aftermaths are still there. So as I said, there are many different types of epigenetic mechanisms. There are modifications on the histone tails that are acetylated or methylated. There are RNA interference from small microRNAs. There's also methylation of the DNA. And we've learned a lot in terms of how those different modifications regulate whether or not a gene is turned on and off. And I'm going to tell you that of all the things that we've learned, we are still only at the tip of the iceberg in understanding the complexity of epigenetic mechanisms. There are now so many more epigenetic mechanisms than we realize that modulate gene regulation. But here, for example, we know that acetylation of histones, for example, will lead to open chromatin genes being turned on. For example, histone DNA methylation will lead to a condensed chromatin where the transcriptional is repressed. When we look in the brains of heroin abusers in the striatum, the patterns that we see in terms of what's going up, what's going down, are really consistent with an increase of histone acetylation with a repression of the histone deacetylases, which would mean, again, this tone of more open state of transcription along some of those genes that we see. And we also see that genes that relate to epigenetic modifications, I'm not going to give you the detailed names, but they regulate often with these glutamatergic genes. For example, this is a GLU8-1, classic glutamate receptor that regulates synaptic transmission. And we see also that the acetylation changes relates to the years of heroin use. And one of the MD PhD students in my lab, Gabor Icahn, who's now actually doing postdoc at Penn, we wanted to see, is it specific? Because I said there are many histone modifications in the DNA. And Gabor was very persistent, which was great. And we tracked, for example, where modifications were on the DNA of heroin abusers. And it's actually very specific, which was very surprising to us. It's where the modifications are, they're predominantly at the region of lysine 27, acetylation, which is an enhancer region. I'm not going to go into detail with that, but just to say that it correlates a lot as well with years of use and inversely correlated with toxicology. So what does that mean? Like I said, we see that there are these epigenetic changes that are very specific in the striatum of heroin abusers. It relates to aspects of changes in acetylation at these regions that are called enhancers that are critical for regulating gene expression. We wanted to just see whether or not indeed there were changes in the chromatin irrespective of what epigenetic modification we were studying. And here we use a technique called assay for transposase accessibility of chromatin, but attack sequencing for just what it does is to give you the overall whether or not and the DNA is open or closed. And you can see which genes this open and closed states are. And the thing that's also great is that it allows you to, we could sort cells, look in neurons versus glia, for example. So one of the things going past this that we saw is that there are indeed changes that you could see that completely correlated with what we had seen when at the gene expression level. That indeed, for example, is again showing you this GLIA1, this glutamatergic genes, where that these genes were significantly changed. And here these are in neurons, and there's an open state in heroin abusers. Importantly for us, the question is, especially in the human, they have a very complex life. As I said, genetics is also part of it and other environmental factors, the polysubstance use. So we try to find out, are the changes that we see in humans also there in our animal models, where we know their history of drug use because these animals self-administer heroin when they push a lever, And one of the things that we could see is that a number of the epigenetic changes we saw in the human brain were completely replicated in the same place in the genome that we saw in the same places in the human. And the animal model helps us a lot because it helps us to be able to now go into seeing whether or not can we reverse some of these changes, and can those changes really then impact on the heroin self-administration behavior, for example. So as I said, one of the changes that we saw very strongly in heroin abusers is this impairment of acetylation. And acetylated histones are actually read by these bromodomain complexes. And these bromodomain complexes, we saw there are a number of them in the brain, and it was mainly one that was changed. It's a BRD4 family. And Gabor worked with Diana, and we've been trying to track down, we're trying to track down which bromodomain complex might be impaired. We saw that it was BRD4 that correlated to the acetylation. Don't worry about that. But the one thing that was important about BRD4s or the bromodomain complexes is that they've been highly studied in cancer. And cancer field has developed a number of medications that have been developed in terms of for different types of cancers, but in particular, they are trying to develop ones that are very specific for the different bromodomain complexes. Whether or not they're succeeding is another thing. But because we had some tools, pharmacological tools, we said, okay, let's try at least one of them. And like a prototypical one that people in the cancer field have been developing is a compound called JQ1. So in using JQ1 to inhibit the reading of these acetylations in our animal models, we could infuse the JQ1 into the striatum and reduce heroin self-administration. We could also reduce heroin-seeking behavior. And importantly, because we're not going to infuse a drug into the striatum of our heroin abusers, when we gave it systemically, significantly reduced heroin self-administration in the animals. So that's one strategy for us in developing, and now taking there are many compounds that are similar to JQ1 that are now being used in human studies. JQ1 has some off targets that we're uncomfortable with in taking it into the clinic, but others are, and those are the ones that we're starting up with. One of the things when we did the attack sequencing to look at open state of chromatin, we were looking initially, we used this strategy to be also unbiased, even though we looked at it to test whether or not the glutamatergic genes and so on were altered in the way that our gene expression predicted. We wanted to see, just on an unbiased manner, where is the most epigenetically disturbed part of the genome. And so when we sequenced the epigenome, one of the things that was clear was that it was really surprising. We looked in the neurons and glia, and of course the cells made a difference, but disease, specifically in neurons, was a high prediction of epigenetic variants, and the gene that had the greatest change was this gene called FIN. And I don't think I would ever have looked at FIN necessarily. And the thing that was fascinating about FIN, and I'm going to go over a number of reasons why it's fascinating, but it also it sits. It's a Src kinase that sits at the postsynaptic density and regulates glutamatergic transmission, in particular the NR2B, also important for glia. And we also saw that these genes had these epigenetic alterations, but FIN was the highest. And it was specifically in neurons where the chromatin accessibility was. It was not in glia. And when you look at FIN, as I said, it's really fascinating because there are two things about FIN that well, many things. One, as I said, it sits at postsynaptic density, important for synaptic plasticity. It also phosphorylates tau. And tau is really critical for, the synaptic dysfunction that's there in Alzheimer's disease, for example. But one of the first things we did was to just say, again, can we replicate this? So we see that on the gene expression level, not only is it at the epigenetic level altered, but the gene expression level also in our rats and even in our cell culture. So we know that it's heroin or opioids that are inducing these changes in the FIN gene expression. We also, because of this interesting regulation of tau, a number of years ago, I had worked with a pathology resident who wanted to get into research, and we said, okay, look at all of these marks for neurodegeneration. And it was mainly to help him get a paper. And we had seen an interesting finding that I had just put down to it being that most likely heroin abusers, they're falling down, they're hitting their heads, probably that may be causing why we saw this. That there was this increase in phospho-tau pathology in the brains of heroin abusers. And now we come back to the fact that we saw it on an epigenetic level. So it can't just be that they're hitting their heads, and that we could reproduce it in the animals. So FIN phosphorylates tau at this this tyrosine 18, and we could see and replicate that they're indeed very specific in heroin. It regulates the tau, and in fact, it correlates only in heroin abusers. The phospho-tau correlates with the FIN gene expression, and the specificity is really important because there are other parts of the tau protein that are phosphorylated by other kinases, and those are not changed in heroin abusers. So there's something very specific about FIN with heroin. So we also could, in our cell culture model, again, replicate it. So you can see that we now have these very significant changes in phosphorylated tau in our cell culture treated with opioids. So there are now a few medications that are being developed that actually targets FIN, and I call compound X. And in using one of these, we could see that in animals that self-administer heroin, if we gave it to them, we could decrease their heroin self-administration behavior. And in trying to develop medications for addiction, we also wanted to be specific that it's not just hitting every aspect of their reward. And if you look, for example, in just food self-administration, it doesn't change their food self-administration. So it's very specific to at least heroin. I don't know for other drugs. Mm-hmm. That's a great question. I'm going to tell you these studies that the postmortem brain collection that I have, the majority of them come from when I was at the Karolinska Institute in Sweden. And I collaborated with my colleagues in Hungary so that we had very similar population. At that time, it was predominantly intravenous. And they are very homogeneous in terms of, at least on both levels, in terms of racial, it's such a very unique population, but it's predominantly intravenous. Now in New York, I have a lot of different intranasal, all this, but it's mainly overdoses that I'm showing you. Mm-hmm. You're welcome. All right, I forgot to say, so thank you. Please stop me, ask questions. Oh, okay, so we have the animals self-administer. We actually even increase their how much effort that they have to work to get the heroin, and so they go up. And so here on day 11 is when we gave it. So the interesting thing about this compound is that it's only effective that day. And when for example, this day when they don't get it, they go back up to it. So this compound has to be in their system in order to be effective. Thanks for, again, reminding me about that. Mm-hmm. We haven't checked that yet, but that's definitely, so I went for the food first, and these are chocolate pellets. So hopefully, but absolutely that's going to be the key thing as you try to develop these medications, that it's not just having these broad effects on reward processing and hedonic processing. So in any case, we are now taking these types of compounds into clinical trials. I'm fighting with a company. It's really tough because when people develop or companies develop a lot of these medications, it's not for the addiction population. No, many of these companies don't want to develop medications for treating substance use disorders, and that is a big battle. And I think we really need to be much more vocal, and it's just very upsetting. So there's a lot of barriers when you find something that works, it may work. I mean, I have no clue. That's why you need to do clinical trials. These are still animals. If it works in the clinic, it'll be great. But it's just trying to convince companies to put in that effort. So, like I said, everybody in this room have to make that commitment to being vocal and trying to help. The resistance in large part is stigma. I mean, obviously, treating cancer is one thing. I will say also Alzheimer's disease as well. The stigma, as many of you who treat people with substance use disorders, they're not the most compliant people. It's challenging. But even though it's challenging, showed the numbers in the beginning, we have, over 500,000 people in the past decade have died. And if we had 500,000 kids who died of a particular cancer at the same time, everybody would be, on this. And so it's just the stigma that addiction has, and that this moral thing. So a lot of the companies, also, they don't think they're probably going to get back their money, because most of the substance use, the patients are on Medicaid treatments, Medicaid-funded treatments, as compared to Alzheimer's and cancers. So it's stigma, it's money that they can make back. But I think that if we can alleviate, and there are a number of preclinical studies that are very interesting that I think need to be tested in the clinic, but I think that there's a lot of resistance. So in any case, for us, this was just like a little summary that for us, what we see is these acetylation that regulate transcription of a number of the genes, and that it changes the accessibility of the chromatin for genes, especially because I'm regulating the transcription in the nucleus. But interestingly, it also leads to these changes that we see in the synapse, especially within the postsynaptic density. And so that's where we're targeting. So here, JQ1 types of medication, but also here, like this compound X type of medication as well. And those are the two epigenetic targets clinically that we're developing. The second part of the research efforts that we're doing in terms of medication development really stemmed from the issue of cannabis use in our country, where we know that there's over, what was the number showed again? We have over, this number is actually wrong, but we have around 30 million people with a cannabis use disorder. And we know over the past decade or so, the ratio of THC to cannabidiol, which is a non-intoxicating cannabinoid in the marijuana plant, that ratio of high THC has gone up. And the issue for us is that, does it impact on addiction? For me, a big question is on the developing brain, because we know that those contribute to aspects of, for example, psychosis. And a lot of long-term studies have shown, the association between early marijuana use and future vulnerability for opioids, like stimulant abuse later in life. And also, the frequency obviously matters. Multiple studies, ours is not by any means, we just add to it. This is an addiction fellow who worked with me, who's now assistant professor at Yale, Anahita, in our inpatient population. Definitely both synthetic cannabis, in particular synthetic cannabis, but also natural marijuana is in our inpatient population is what really we see driving a lot of the psychosis and agitation and aggression in our inpatient population. And the changing demographics of now this pendulum swing of cannabis being terrible and now cannabis being great. There are a lot of states that have legalized marijuana, and there have been initial reports that there have been this reduction in opioid use in some of these states. For example, the number of prescriptions are now reduced for opioid painkillers. There's a reduction in opioid overdoses and even reduced positive opioid toxicology screens in people with fatal car accidents. These are not, I mean, we'll see if this is really real, because these numbers may be reflecting many different things than just marijuana per se. But what it meant for us, we have been looking at the developmental effects of marijuana for many years. And in our animal models, we mainly have used THC, and where we would expose either during prenatal time periods, because we also have a population of women who smoke marijuana while pregnant. And in one study, we've studied the fetuses of the aborted, some of the women who have had abortions, we've studied their fetuses, and others we've studied the behaviors of their offspring over time. And we also looked at the adolescent exposure, and where we can also look into adult behavior and the molecular studies. And the one thing that we always saw, oh yeah, I'm going to take that back. Always is a terrible word, because we don't always see it. It does depend on behavioral traits. But in general, if you give THC during adolescence, you normally see that this increase in heroin self-administration when they're adults. And we also see that there are changes in, for example, social stress. So if you social interaction with animals that were adult animals treated with THC, they showed this impairment in social interaction. They also showed that after these social stresses, and social stress, actually, we've tried many stresses, because there is something that we see, stress is really key with developmental exposure to THC. But after social stress, which is one of the biggest stresses that we've seen, they will even change their reward sensitivity, and, for example, self-administering more food. And each time they get exposed to these types of social stresses, it enhances some of, in certain doses, their reward sensitivity. We also looked at, and because of this gentleman in the room, also looked at the prefrontal cortex. We focused a lot on the striatum because of the key role that the nucleus accumbens, for example, plays in goal-directed behavior and reward, and the dorsal striatum for habit formation. But the fact that the prefrontal cortex was one of the last regions of the brain to develop, and all of the aspects of prefrontal cortex with psychiatric vulnerability, we started looking at the prefrontal cortex, and even, for example, looking at the morphology of these, like, for example, pyramidal cells. And in adult animals that had adolescent exposure to THC, you see that the actual length and so on of the neurons are normal, but the complexity of the neurons are very different. So here, this is in green, in just showing that there's impairment in both the apical branching and the basal branching, just again, just showing that the complexity differs. We also, for me, a major issue was, it worked before, it changed it. Okay. It was working before, but it doesn't matter. It's a grant access? Okay. So I wanted to know, we see that the morphology is different. What is the molecular makeup of these cells? So we use a laser, and we went in a laser and captured these pyramidal cells so that we could then go and do molecular screening of these cells. And so what we see when we capture and sequence the gene expression in these cells is that there are two predominant dysregulations that are there. One deals with really strongly the cytoskeleton, the dendritic development, which fits in line with the morphological changes that we see. There's huge changes in gene networks related to, like I said, morphogenesis, the projection of these cells, and epigenetic modifications. There's huge histone changes in terms of gene expression. In fact, there is a very strong correlation between the genes involved in these epigenetic modifications and in dendritic regulation. So again, these are vehicle animals on the right, and this is the relationship between adult animals with adolescent THC exposure, that there is this reorganization of the epigenetic landscape and but it's in relation to genes that are part of synaptic plasticity. And in order to just a quick thing in terms of, it was interesting for us because what does this mean for aspects of the human brain? So we looked at databases that had human gene expression from the prefrontal cortex. And what was fascinating is that the dysregulation that we see in the animals correlates really strongly on the same gene networks that have to do with dendritic branching and so on. And this is the schizophrenia common mind. So mainly in schizophrenic subjects, we're seeing a very similar pattern that we see in our animals in these specific cells that deals with the structural development, and it overlaps with these epigenetic disturbances. But as I said, we were studying THC for when we talked about cannabis. And we know that there are many other cannabinoids in the marijuana plant. In fact, there are over 500 chemicals, and there are about 140 cannabinoids. And one of the cannabinoids that, as I mentioned, that had started to decrease over the past few decades is cannabidiol. And a number of studies had started to see that, in contrast to THC, which we know increases psychosis, and for us at least, we saw these dysregulation in regard to synaptic plasticity. Studies had started to see that CBD actually decreased psychosis. So a question for me in terms of this yin-yang, that many of the ways in which THC or CBD kind of counter some of the things we see with THC, I wanted to check out opioid addiction. So we went to our animal model, and as I said, we know that when you give THC, you then see this increase in opioid self-administration. And we actually saw a very different pattern when we gave CBD. In fact, I thought that we were, well, that's why you do research. Your hypotheses are usually wrong. But I thought that we would see something, either it would be a little similar, or that it would change the heroin self-administration itself. But it doesn't. When animals self-administer the drug, they get a cue. Here for us is this light cue. So when they see the light, they know they're getting heroin. And after a while, if you just show them the light cue, they'll start pressing away to get, trying to get the drug, even though they're getting no drug. And that's an aspect we interpret as drug-seeking behavior. And that's where CBD really was very strong. It decreased the heroin-seeking behavior. And importantly, it did it, I don't know if I have, it did it even 2 weeks after their final CBD, it still had this impact. So because of a lot of the fact that we have seen these glutamatergic impairments, we know that there are these glutamatergic impairments in heroin abuse, and we looked at CBD's impact on a number of genes. This isn't sequencing. This is just like hypothesis-driven, and we didn't see changes in our normal dopamine cells. But what happens is, for example, in animals that self-administer heroin, there's a disturbance of glutamatergic systems, and CBD normalizes it. It also normalizes. In heroin abusers, you also have impairments of the natural endocannabinoid system, and here's the CB1 is increased, and CBD brings it back down to normal. So it's normalizing a number of the genes relevant to synaptic plasticity. So we decided, let's try to be crazy and go into the clinical lab study. So we still didn't know at that time. Actually, many people around the world were still trying to figure out cannabidiol in large part. But I decided that instead of figuring out all the mechanism of action of cannabidiol, let's first see if what we saw in our animal models were really relevant to what we were seeing. Were really relevant to the human condition. So for the young people in the room, and even for the old ones, we've all gone there. First, you had to get the FDA approval, which took a long time. IRB, get money to support it, get a company here, back to the company. I had to beg GW Pharmaceuticals, I was on my hands and knees for 6 months with them. And then the lawyers between their group and Mount Sinai, a year later, it was crazy. So finally, we were allowed to do our clinical study. And, oh wait, I should say, we did take like the first, after all the stuff with GW, then another year to show the FDA that cannabidiol was safe. And safe in the sense of if, for example, someone with an opioid use disorder relapsed while on cannabidiol, that there would be no negative drug-drug interaction. We did that safety study. They forced us to use, I had proposed using morphine. They forced us to use fentanyl, which, you know, reviewers were really upset with us, but that was what the FDA wanted. We monitored them, obviously. And so we did all the safety and pharmacokinetics of it. And so then we finally got to be able to say, okay, does this 2 years later, or probably maybe 3 years later, does this work? And we focused, because of the animal study, we focused on craving. And craving is a key aspect of relapse. And we know that, you know, for a lot of patients, the environmental cues have a really big impact in their motivation to start using the drug again and the relapse of it. So I'll talk about I'll go back with, I decided I didn't know really where to start because there weren't many studies at that time being done with cannabidiol. So I decided to just do what we did in the rats. So people ask me all the time, how did you come up with this? I just did what we did in the rats. And basically in the rats, we had looked at the acute effects. So immediately after that time period, actually, I did make a little bit longer in the humans. And then we looked 24 hours later because we saw that still had an impact then. And we had given the rats actually the first time 3 injections of CBD in 3 sequential days. And then we let them come back like a week later and we looked at after their final CBD. And we did 2 doses, again, the dose range that we had studied in the rats. And that we know from other clinical studies with CBD was below, people had used even up to 1,500 milligrams of CBD. And the first study, GW made me capsules. And so this is what, excuse me, Sharon was a clinical coordinator at Celsus, is really phenomenal addiction medicine doc, and it's been fun working with him. So what we saw that when we showed people the cue, we brought them in the lab, and we showed them that, oh, sorry. This first study, we also learned, so we did this through the clinical research unit at Mount Sinai, where we had to start just for certain practical things on a Monday morning. So we'd line everybody up, everybody wanted to be part of the study. This became a pilot because the majority of people relapsed before we could even give them the first CBD dose. Mm-hmm. No. Yes. Yeah. So that's what I said. So the IRB, exactly. So exactly. Oh, right. In terms of, exactly. So we weren't thinking, we were naive. We still kept the same thing even for the second study, but we were definitely naive in terms of thinking, oh, everybody will be off. They really want to be part of it. And like I said, the majority of people relapsed. So it was not well thought out. And so this was our first step because we just wanted to be as pure as possible and didn't want other opioids there. And, so like I said, definitely naive. But in that naivety, we still were able to get a few things. And the thing we were able to see that showing people a heroin cue increased the craving in people who had gotten the placebo, but the CBD reduced that. We also saw that we didn't test in our animals at that time that actually anxiety is a huge factor. So the increased anxiety and CBD completely blocks that. And like our animals, when we brought them back a week later, their general craving CBD was also decreased. So like I said, because of all of that, we repeated the study. This time around, I've now become the director of the Addiction Institute. I'm able to have more control over our clinical populations and where things are done. And so we redid the study. There's still a lot of flaws in a number of things, and again, the growing pains of all of this. But in general, what we've seen has been fascinating. And it's fascinating because it replicated that first pilot. Again, I forgot to say these are double-blinded, placebo-controlled studies, and acute first exposure to CBD. So when they get the heroin cues, there's an increase in craving, and CBD decreases it. 24 hours later, we still see that trend. At 800 milligrams definitely is more efficacious. And a week later, there's a significant reduction, even though the in-lab craving over time does habituate. We've shown them paraphernalia to try and increase, but it's still habituate. But nevertheless, CBD still decreases that. We, again, replicated that anxiety is a key factor. There is a increase in the heroin cue anxiety, and CBD reduces that. And again, we see that 7 days later, CBD is really still effective at reducing their anxiety. We've measured also cognition on the last day of the CBD treatment, the third day, and we didn't see any significant effect on a number of the cognitive measures that we had. Whether or not we needed more time for the cognitive effects to show itself, or just the tasks in which we had used, those are things that we're now going to work on. But the protracted effects of CBD, to me, are definitely fascinating. And remember, I told you, and I showed you that epigenetic drug, that it had to be in the system for it to work. And CBD does not appear that you have to have it every day in the system for it to still be effective, which I think is really important in trying to treat addiction populations. We also measured a number of physiological marks, like their heart rate. And so some people would tell us, oh, those cues are ridiculous, yet still their heart rate would go up. And CBD decreased the cue-induced craving, but we also saw that so the drug cue increased cortisol, and the CBD really decreased their cortisol levels to the drug cue. So overall, for us, we see that cannabidiol does show promise for treating opioid use disorder, and in particularly, it's really relating to craving and anxiety. And in our animal models with our epigenetic targets, we never really looked at anxiety, and even though we did see drug-seeking behavior, I'm going to end now, but there are different things that we see with our epigenetic targets where they specifically reduce drug-taking behavior. And it does impact on drug-seeking later. But CBD mainly reduces drug-seeking behavior. So there are going to be differences, I think, in the mechanisms of the medications being developed. And I do think it is important to have different strategies, because substance use disorders, I mean, you need many different types of medications for treating different phases of the substance use cycle. So for us, clearly, on the epigenetic reorganization in the human brain, I think lends itself to targeting for medication development. This is actually an actual heroin-induced impairment on the dendritic impairment related to opioid use. It changes the structure of the brain that I think that we hadn't really appreciated before. We see these changes in, it's not neurodegeneration like Alzheimer's, because not other marks that are relevant to Alzheimer's pathology weren't changed. It's very specific to this phosphorylation of tau that I think links back to the glutamatergic changes at the synaptic level. But cannabidiol is also interesting because it seems to be able to normalize a number of the glutamatergic dysregulation. We haven't tested CBD on whether or not it can change the morphology, morphological changes that we see that opioids do, and that's something that we're working on. But these two strategies, I think, have promise, and we'll see whether or not they do by continuing clinical trials. But I just said, I want to thank a lot of the people, Anahita Basirnia, and Claire Mann is a medical student who has been phenomenal in our cannabis psychosis studies, which I didn't really go into so much today. Michael Miller, who did a lot of the adolescent THC studies, and with Ben Chadwick. They hated me in the beginning when I said, go and pick up every cell. And then when they realized it could be like a laser game, all the guys were in for it. So, it depends on how you motivate. And Gabor and Noel for a lot of the postmortem human studies, and a great clinical, our coordinators and our technicians are amazing for us, and obviously NIH. And I'll take any questions. Thank you.
Thank you, Yasmin, for that outstanding talk, which told us not only about basic mechanisms, but maybe 3 different approaches to therapeutics, at least, that have come from understanding those mechanisms. So I'd like to now open this up for questions from the audience.
Yasmin, about the CBD, do you think neurons are still the major target? And how about the receptor? Is it still CB1 or?
Yeah, so we know that CB1 is not the main target for CBD. It is, everyone is trying to figure out why is CBD working. And it appears to be a negative allosteric modulator at the CB1 receptor as compared to THC, which is a partial agonist. The GPR55 seems to be an important target for CBD, but also the 5-HT1A and a number of receptors. So one of the things, actually, I like CBD for the fact that it is not a potent at any at any of the receptor targets that we see. And that might be something I think our field has to start thinking about. We see that a particular transmitter system is impaired, so we said, let's get a big hammer and block it. And CBD seems to fine-tune multiple systems. It's not powerful at any one system. And I think that that is something, as I said, we know that a lot of psychiatric disorders, it's not one thing that is off. There are a number of things that are, they're interactive systems. And so my thought about CBD is that because it is a fine-tuning a number of these systems, it's bringing things more in a homeostatic way than just as an antagonist or as a full agonist. The first part you asked was
neurons are still.
I don't know. I don't know. We're doing the same things now in sorting different cell types, following CBD, and doing sequencing. So we'll see. But CBD, like THC, actually does have important immune function, and I think the glia will be an important part of that. I don't think that CBD's effects are neuron specific, but that's just a hypothesis based on other things that we see.
Very excellent presentation, thank you. My question is more clinical, given the high comorbidity with opiate use disorders and depression and anxiety, and most likely the involvement in not only the glutamatergic but dopaminergic, serotonergic system. Until there's new drug development available, I'm wondering if a patient comes in, should we be treating them, starting them all at once on a not only buprenorphine but a dopamine agonist, SSRI, GABA medication, and because of its extensive ramifications, at least eventually, start those all at the same time? Or
I think that that's what's been done, right? Clinicians patient comes in, you see this, they have depression, you add this antidepressant, you see this, mood, you add, so you do play the pharmacist. And I think that that's one of the reasons, like I said, perhaps CBD might be interesting. And it doesn't have, it's not blocking anything. So if someone does take heroin, they're gonna take heroin. CBD is not gonna block opioid receptors. They're still gonna feel that reward. It's just that they're not going to crave heroin as much to then go and take it. So I think instead of giving multiple drugs, it would be great to have a medication that in a way kind of does some of the things that you're saying, the serotonin, the dopamine, and so on. And that's where I see that CBD might play a role. Also, when we look at it across a dose range, and other people have studied high rate, we don't see serious adverse effects. And I think that that's also something very important. A lot of the other medications have a very, very narrow safety profile and how they interact with other drugs. So it, you know, I don't know, and CBD might then turn out not to be effective, but that's why we need more clinical trials. And I think many groups, I hope more groups start studying it so we can see what's maybe real, that we don't need to add 5 other medications to the buprenorphine. We can add, you know, CBD or something like it.
Yasmin, first part of your talk, you mentioned the FIN gene. So what about the FIN gene in the cannabinoid scenario?
So we do not know FIN in the cannabinoid scenario. So most of the things that we've done, as I said, we're doing the sequencing. So we'll see if the FIN gene and the FIN network turns up. It will be fascinating. Yeah, but I can't, yeah, no. But FIN is really exciting and interesting for us.
Thanks for the wonderful talk. My question is concerning the first part of your talk, the heroin brain and the epigenetic regulation. Your analysis more at the tissue level. You mentioned the nucleus accumbens. I wonder whether maybe you've already done it or planning to do it, if you'll be able to refine at the neural circuit level where those changes are happening. For instance, is it glutamatergic receptors in the postsynaptic elements of the medium spiny neurons, or in the ventral tegmental area dopaminergic neurons also going to the nucleus accumbens? Yeah. It would increase the detail of the picture where the mechanism is.
Yeah, so in the human brain, it's been challenging. So we've worked for years trying to identify, at least in the striatum, a marker that can give us, dissociate the different types of medium spiny neurons, for example. We cannot still find a marker in the human brain that can help us with sorting with that. We can only do neuron glia. For our animal models, though, so now we're using retrograde labeling and then taking those cells that project, for example, to the VTA versus those that project to the pallidum. And also, because we see this really strong effect in the pyramidal cells that project to the amygdala, and also project to the striatum, we're also doing that loop. So in the animal studies, we'll be able to tell you more refined in terms of which synapses may be more vulnerable. Yeah. 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 Yeah, Tani Rahman, and she's been developing the RNA scope in our lab. Either the morphological intactness of the human brain still has not been beautiful. It's still our animal models for that. So, but we hope that she'll work harder. No, she's amazing. She's amazing. But so she's trying to develop those things because I still want to see it in the human brain too. But that's the thing that's been challenging.
Yes, and I'm going to ask a question. So I know very little about addiction, but Koob has drilled into me that addiction is not the disease we once thought it was, and that we should be focusing more on the dark side of addiction. And you look at, on the one hand, most of the biology you presented to us was at the level of the striatal and prefrontal cellular and molecular changes, and yet the symptoms that you were focusing on, in your, particularly in the CBD side, were on the level of craving and anxiety, which sound to me much more like the
Oh, where's that? Yeah.
the dark side, and even cortisol levels. So is it possible that you'll
I was going to show you some data. Yeah.
okay, good. Learn more about these mechanisms, looking at structures outside of the traditional reward systems, more at levels of the extended amygdala or other circuitry relevant to anxiety and craving.
Yeah. If that young man comes back, I will show you some. Oh, he's here. I was trying to get into this. Oh, no. What happened? I hope it's still there. If not, I have my computer. I can just quickly show you. Look at, okay, so George is correct. And one of the things that I think is important, and that's why the question about the anxiety is an aspect that I think people forget to realize. And again, I sound like a commercial for CBD, and I'm not really, but you know, it is interesting that it had such strong effects on anxiety. So is it the anxiety or is it the craving? I think it's a combination, and anybody working with substance use disorder, I will take whatever that reduces, wait, I'm just trying to, so it was just to show that one of the things that people, wait 1 second. Oops. I completely agree with George. So the amygdala is a key place that we also see significant dysregulation in people with heroin use disorder, and oops, it will come. And so the one thing about the amygdala, though, is that I hate it, is because it has like 15 subnuclei, and so many years ago when I was what was I? I was a postdoc at NIH, and I started saying, let me look at the amygdala, because I had gotten access to the postmortem material, and one of the things that I saw was that dynorphin, which is an opioid peptide that relates to negative affect, was significantly altered in the amygdala, in a particular brain region especially, in people who were diagnosed with major depression or bipolar disorder. Didn't see it in schizophrenics. I tried to convince people over the years in my group to study this, and nobody did. But I had seen something where, and then Sarah Ann Anderson, an MD PhD student in my group, who, well, a long time ago, she's now in Columbia doing her, well, she's past residency. But in any case, she replicated what I saw. So in people with major depression, we see the same dynorphin impairments. But we also saw it in people with heroin use disorders, and also people with cocaine use disorder. We only published on the opioid. The problem is that it was in this region that nobody in the animal field ever studied. It's in this region called the periamygdaloid cortex. I'm trying to like, this area up here. People study the central amygdala, people study the basolateral, but in the human brain, this is where we saw very much disrupted in people with heroin use disorder. So we tried to figure out what it meant. So we used the technique of going in and with a viral vector that regulated the firing activity of these dynorphin cells. And it was really crazy. Just changing the few of those dynorphin cells, their activity, recruited this network, which is the extended amygdala. And it goes up in this foot that you see is the cingulate cortex, where, you know, like deep brain stimulation for people with intractable depression. So it is definitely the brains of people with heroin use disorder, but I'm going to say also cocaine. It didn't seem to be specific to at least heroin or cocaine. And this neural network, this had extended amygdala. And the thing that was fascinating is that by changing the activity of those cells, we could change their peripheral cortex levels. We could change their sucrose preference. We could change their hedonic state and also change their depression-like behavior. So absolutely, the dark side, quote unquote, the neural circuits in the brains of heroin abusers absolutely shows that the amygdala is impaired, but it's not necessarily their unique circuits. But it also brought up something to me that's fascinating and scary at one sense as well. A goal for me is that since we can target this cell population surgically, we could go in and give put a vector in that changes the firing activity, because it was a receptor like these dreads where they're designer receptors, and you give them an inert ligand that it doesn't activate. Well, now we know it does, but it doesn't activate. It doesn't activate. In theory, we want to develop one that doesn't activate any endogenous receptor except the artificial one that you've put in that only stimulates this. But it tells me that we actually have very little clue about the anatomy of the human brain. These few cells that are changed with this dynorphin gene could change your peripheral cortex levels. It could change your depression-like behavior, and no one studies them. So, I think that we're still in neuroscience very much naive about how far we've gone. We've gone very far, but we're still very focused, all of us, on the accumbens, on the dorsal striatum. We're still very focused when these, the complexity is much greater, much greater. So I do agree with George, and the fact that we saw, like I said, that CBD is mainly having, I think, a very strong effect on that negative component. It didn't change depression. So our depression scores, well, again, to the point earlier about who were the patients, they weren't really showing depression. So I don't know, the clinical studies done with CBD thus far doesn't show depression. Like some animal models of depression did show CBD is effective, but in clinical studies, I have not seen that CBD. It's mainly on the anxiety spectrum. Sorry, that was a long answer.
I'm going to ask you to change gears a little bit and tell us in some more detail about your experience working with the pharmaceutical companies and the FDA, and to see if through that experience, is there any advice for somebody on the playbook for how to get a clinical trial conducted or how to get industry more excited about these kinds of projects looking forward?
Yeah. One thing about the opioid epidemic, it did change in the past few years how industry is looking at it. So now, for example, naloxone, sadly, they increased the price ridiculous amounts because it was now being handed out to everybody, first responders in schools. And so now pharmaceutical companies start to think, okay, there may be something here. And there are now generic naloxone that's being developed. So I think that there is now a little bit more sensitivity, and especially since a number of the people who develop opioid use disorder, this thing that, it's people who have bad morals and so on, and you saw that it was people who were given the medication by their physicians so that it wasn't any fault of their own, quote unquote. But so I think there is some push for the pharmaceutical industry. I think it's NIH. So NIH has joined in partnership with a number of companies, and so perhaps that might be one of the first places to start. So like NCATS, to see which companies have partnered with NIH in developing medications, in potentially developing medications for any psychiatric or drug abuse disorder. The FDA is more open. Absolutely, I think that they themselves were learning a lot, and that many people think that you can't treat addiction. And I think that's the whole thing. If we get people away from that mindset, FDA included, and these pharmaceutical companies, that as the science improves and as we understand more about the neurobiology of disorder, that we can indeed use that to go into clinical trials. But many people know that the timeline to go from developing or finding a target to then having something phase 3. I'm not interested in phase 3, for example, but somebody else, I hope, would, if all our results and other people come and show this, they would take it to a phase 3. And that's what the companies using. GW Pharmaceutical, I had to go on my knees and beg them. And they definitely did. I forgot until the second time around. Now they had Epidiolex in the US with a distributor. So I decided to just use the Epidiolex that's being used for kids with epilepsy with Dravet's. Their clinical studies, they targeted orphan disease. And so they didn't need that many patients. And now they're one of the first, in fact, the first company to have a FDA-approved cannabinoid. That's going to change the scheduling for cannabinoids, and that will also make it easier to develop clinical studies, because it is challenging when the FDA, the schedules of these drugs. I mean, cannabidiol, I had to have, we had to have a guard walk my coordinator to the pharmacy. It's not addictive. It has no rewarding properties, and just because it's a cannabinoid. Once we change those things, so there's a lot of things to change. The FDA has to change scheduling. I think that they're definitely more receptive to addiction-related therapies than they were in the past. And I think working with NIH to as you develop your protocols, or work with the companies as you develop your protocols. But I think we have to do it. So I don't think that we have a choice. I just think that we need to be much more vocal and visible. I think addiction, except for the neurobiologists, you talk about George and a lot of the, and Nora Volkov and so on. I think on the neuroscience level, the basic science, we are very visible, and it's considered a very strong neuroscience field. But clinically, the people working with people with substance use disorders, they're also stigmatized in part. And I think that many people kind of went to the side and didn't want to be seen. I think that we, as a field, just need to be very visible. And I think that will change a lot of the perception of, yes, substance use disorders can be treated and managed. Yes, you can use science to develop medication. Yes, you can conduct clinical trials in a reasonable time frame that can be, provide medications that companies can commercialize and make money. So again, a long answer, sorry.
More questions?
I've always been fascinated by your adolescent data, and what I wonder, because it seems like it's on the low end of exposure, right?
Yep.
What I'm really curious about is if you ever had any other studies with higher intensity exposure in any way that you looked at the same heroin endpoints.
Yeah, so definitely when I started, I wanted to go low, and because, like I always tell people, you can blast anything out of the brain and see a response. And over time, because we see for a lot of the social stress, it's the higher dose THC. So we have a lot of studies now, and thank you for analyzing some of our toxicology samples. And we see that the higher end concentration of THC really does impact on the stress responsivity. And so Jackie Furlan and I showed a couple of things just in terms of social stress and reward sensitivity. So we do see that dose there does make a difference. And we're bumping up the dose of THC. But it is important to emphasize that even the low dose THC have these long-term effects. And that, our studies, we even saw it transgenerationally. So, you don't need this high THC, but the high THC does acutely change a lot more the stress response.
Other questions for Dr. Hurd? Well, I'd like to thank you again, Yasmin, for an outstanding talk. Clearly, lots of interest and everything from the molecules to how we get the FDA and drug companies to give us this compounds to study. I want to first, can we give Yasmin some applause for?
All right, thank you.